Hi, I'm Kylie Pfeiffer, and lucky enough to be speaking with Dr. Scott Murray from the University of Washington today. He studies the neurological basis of visual perception. Dr. Murray, thanks so much for taking the time to be with us today.
Thank you for having me.
So, could we start out by you just walking us through the steps of visual perception and how that works?
Sure. First, light enters our eye and gets transduced by our retina. And then from the retina, it makes visual information travels to the cortex. It makes one stop in a structure called the lateral geniculate nucleus in the thalamus, and then projects directly to the cortex, to an area that we call V1, or primary visual cortex. Then from V1, visual information travels to other visual areas within the cortex, labeled V2, V3, and up into the parietal cortex, and also down into the temporal lobe as well.
So where then does perception start in that pathway?
Well, that's a really good question. So if there are upwards of 30 different visual areas, I think it's fair to say that not any one of those areas is exclusively involved in perception. It's probably a complex interaction between all of those different areas. And it really depends on exactly what is being perceived. So for example, we have specialized areas of the cortex that process, say, motion. So if it's motion that's being processed or perceived, that particular area or group of areas is going to be highly involved. Similarly, if we're perceiving things like faces, areas that are specialized for processing that kind of information are going to be involved as well. So it really depends on the kinds of information that are being processed in terms of which visual areas are actively involved in perception. And
so is all the visual information that's coming into our eyes present in all of those areas, or does it kind of get split up?
Well, at least in the early stages of visual processing, it's likely that they're probably all equally involved in any given visual perception. But the higher visual areas, areas that are specialized for specific kinds of visual information, whether it be faces or motion, are fairly selective for the kinds of information that are present in an image.
So information comes in through the retina and goes through this stream. And I know that some terms in cognitive neuroscience are top-down and bottom-up processing. Where would you say that happens, and what are those different functions?
Well, that's a really excellent point, because the way that I've described it and the way that it's often described in neuroscience textbooks is this idea that visual information just sort of cascades in this feed-forward manner through the stages, through the hierarchy of the visual system, and then suddenly we recognize what we're looking at. But in addition, there are massive feedback connections within the within visual cortex. So any two areas that are connected in a feed-forward way also are connected in a feedback way, so that visual information can actually travel backwards through the visual system.
So the term feed-forward then refers to bottom-up processing, and feedback is referring to what's known as top-down processing?
That's exactly right. So, and in terms of what feedback processes are thought to be used for, one well-known example is visual attention. So we can modulate activity in lower visual areas through attention, and it's thought that the feedback is important for that. But in addition, feedback likely is very important for affecting sort of contextual modulations of activity in early visual areas. So surrounding information, for example, can have a large impact on the neural processing in early visual areas due to feedback projections.
So then what are the main characteristics of this visual processing?
Well, visual perception is often thought to be an attempt by our brains to characterize a pattern of light falling on our retina. That is, we want to try to measure precisely what's impinging on our retinas. But in fact, it's probably more accurate to describe visual perception as an attempt by our brains to figure out what out there in the world caused the pattern of light on our retina. And it's a really important distinction to make.
So then is that a distinction between the distal and the proximal stimulus?
That's exactly right. So the proximal stimulus is the stimulus that's actually on our retina, that is the light impinging upon our visual system. And the distal stimulus is the thing out there in the world that caused the stimulus. And it's really important to think about in terms of, say, brightness constancy, for example. So you take some object and could be a white cat, for example, out in bright sunlight. You take that cat, you put it in a shadow. The amount of light reflecting off that cat is now equivalent to, say, a black cat, but you would still recognize that cat as being white.
And so constancy has other forms too in our visual perception, right?
Say size constancy as well. If I stood up, you would recognize me as 6 feet tall, no matter if I was standing here or at the back of the room.
Even though the proximal stimulus was changing on my retina.
Right.
So how has our understanding of visual processing changed over the years?
Well, from a neuroscience perspective, a lot of emphasis has been placed on really the proximal stimulus. That is, we present a particular visual stimulus in an experimental setting, and we ask how neurons respond to that stimulus, or what the fMRI response is to that stimulus. But when we start thinking about the distinction between the proximal and distal stimulus, it becomes more appropriate to ask how the visual system transforms the proximal stimulus, that is what's on the retina, into an understanding about what's out there in the world. So I think in the last 5 to 10 years there's been this increase in awareness of thinking about how that transformation occurs from the actual input to the visual system to its transformation to what's out there in the world.
And where is it thought that that kind of takes place in
Well,
the brain?
traditionally it was thought that that sort of transformation would occur in higher stages of the visual system that would code, for example, an object's identity. Early stages of the visual system were really thought to reflect the input at the retina.
At a very raw
level. And what's starting to emerge now is this understanding that multiple visual areas, even very early visual areas, actually are involved in this transformation from the proximal to distal stimulus.
So in most psychology textbooks, they mention that this constancy happens in higher visual processing areas. Is there any evidence that it happens earlier?
There is actually. So I've been doing some recent studies looking at size constancy. So for example, if you have two objects and one is perceived to be further away than another, but they actually project the same image on your retina, same size image on the retina, you perceive the one that's further away as larger than the one that's closer. For example, in this example here, what we have here is just a simple hallway scene, and these two spheres are actually the same physical size. They occupy the same number of pixels on the image, have the same retinal projection, but almost everyone perceives this sphere that's perceived to be at the back of the hallway as larger than the front of the hallway. And so what we've been able to do using fMRI is measure the retinotopic maps in early visual cortex. So early visual cortex has this sort of convenient organizational structure such that nearby areas of the retina project to nearby areas on the cortex.
And that's what's meant by a retinotopic map, correct?
That's right. So for example, if you were to keep your eyes fixed and I shine a very small focused light on your retina, it would activate a very focused area on your visual cortex. So for example, with these images here, each one of these images is going to activate a portion of your map, and we can compare the size of the area activated to the front sphere versus the back sphere. Now, based on the traditional way of thinking about the visual system, that these early visual areas just represent the retinal information, because the retinal information is equivalent for those two spheres, you would expect that the representation in early visual areas would also be equivalent. But actually what we've found is that the back sphere actually activates a larger area even in the very earliest stages of visual cortex in V1.
Really?
It's important to realize, though, that the context in this particular image, the 3D structure in this image, is not being extracted in V1. So when I mentioned earlier that there were areas in higher visual cortex that are specialized for different kinds of informations, whether it be faces or motion or whatever, there are areas in higher visual cortex that are specialized for extracting the 3D structure of scenes. So what we actually think is going on is that these higher visual areas are extracting the 3D scene structure, and that's being fed back through those feedback pathways that I talked about earlier into V1, and it's after that feedback happens is when the rescaling or the remapping in V1 occurs. So if you separated the representations in time, so for example, there could be a veridical representation of the retinal information. That is to say, that in V1, say early time windows or early stages of processing in time could represent the accurate retinal information, and then later stages then integrate the 3D context and rescale it in the direction of its true environmental size constancy direction.
So now, as far as timing is concerned, you do your studies through fMRI, correct?
That's right.
And fMRI tends to not have very good temporal resolution or timing. So how can you answer
Right.
those questions?
Actually, it has lousy temporal resolution, and if we just use fMRI, we'd not be able to answer that question. So what we've been doing is actually using scalp recorded electrical potentials, or EEG, or ERPs, with a very similar stimulus, and asking when temporally do we see an effect. And actually what we're seeing is something like I described, so very early time windows in the EEG response that we believe are that are originating in V1 reflect the true retinal size of the objects, and then later time windows reflect the perceived size. What it demonstrates really is the overall importance of just of that transformation from the proximal to distal stimulus. That's what the visual system is trying to do, and it's trying to do it from the very earliest stages of the visual system onwards.
In a lot of your experiments, you're using actually two-dimensional stimuli that maybe has the illusion of being in 3 dimensions. What happens when we add in stereoscopic and other 3D visual cues to depth?
Yeah, that's a great point. So it's really just sort of an experimental convenience that I use actually 2D stimulus in which you perceive to be three-dimensional. I think adding more cues, whatever those cues happen to be, if they happen to be stereoscopic, is just going to make the effects even larger and more compelling. So for example, if we take this example that we looked at earlier, even though everyone will perceive these two spheres as being different sizes, if you make the cues more realistic by adding stereo, that effect becomes even larger. So I would presume that the effects that we're measuring, whether they be with fMRI or ERP, would also become larger as well.
So what are the limits to this scaling that goes on?
Yeah, that's a good point. So if we keep adding more and more cues and the effect gets larger and larger, is it the case that we could activate twice as much cortical area in V1 for one object versus another? And there are a couple points to consider there. First, size constancy as a perceptual phenomenon is not perfect. So for example, even though you can recognize me as 6 feet tall when I'm close versus far away, you still have access to the fact that the retinal size is changing. So if you look at a person who's across the parking lot, for example, you can recognize that they occupy a fairly small portion of your retinal image. And then the second part of that question really is how much these maps can change, and that we don't know. We're actually adding more and more cues to see at what point this phenomenon breaks, say in V1. And sort of what I suspect is that we have multiple visual areas. We have V1, we have V2, V3, V4, and I would expect that at some point V1 is no longer going to be able to rescale its representation, but possibly higher visual areas would start to reflect larger and larger changes. But that's for future studies.
Now, what has the response been from your peers, since this is kind of really going against what has been traditionally thought V1 was doing?
Yeah, it's going against what was traditionally thought V1 was, how V1 works. It went against the way I thought V1 worked. So for example, this size illusion experiment really started out as a control experiment where I was going to set out to show that at the level of V1, that these two objects would have the same representation. That was the point of doing that experiment, but in fact, what I found was that it actually reflected perception, which was surprising. And understandably, it's been met with sort of a mix of excitement because it's something new, and an understandable amount of skepticism as well, because what it implies are some fairly large changes at the level of individual neurons. So it's implying that in some circumstances, so for example, when the sphere is small, that neurons aren't responding, and when the sphere is big, neurons are responding, which is really a change in the receptive field properties of a neuron at very, very early stages of the visual system, which is surprising.
So what do you think needs to be done for this to be a generally accepted idea of how V1 works?
Well, I think what we really need to do is get data at the single unit electrophysiological level. That is, what are single neurons doing to this perceived size difference? Because it implies that receptive fields are changing. That is, in some circumstances they are responding to a visual stimulus, and other circumstances are not. So we have to actually go in and measure single neuron responses in V1.
So do you have a position then on the two opposing theories of one being more problem-solving way we process information versus the Gibsonian approach to bottom-up, that our visual field has all the information we need?
Right, so one might be tempted to think, based on these results, that V1 is reflecting the size illusion. You might think that, like we kind of mentioned before, that V1 has done everything, that all the information is there, present in the image, and boom, it's done by V1. But actually, it's important to remember where the source of the information is coming from to mediate the effects in V1. And I really think that it's being derived from higher visual areas that are extracting the 3D contextual information. So I think it's actually more in line with sort of the Helmholtzian view that of an inferential process, that the visual information is trying to infer what's out there in the visual world through the use of contextual information. And it's that inferential process that's being reflected in the activity patterns in V1.
So in your opinion, what do you think are some of the main big unanswered questions that you'd like to see answered in this area?
Well, we have a pretty good understanding of how lower visual areas work. So for example, we think we know generally how V1 works in terms of what kinds of visual information it responds to. The fact that it is modulated by contextual information is surprising, but overall we have some idea of how it works. We also have some idea of how higher visual areas work, such as areas that are specialized for processing faces or locations or motion. But what we have a really poor understanding about are those intermediate areas, those areas between V1 and the higher stages of the visual system. So for example, we have very little understanding of even how V2 works, the very next stage of visual processing after V1. And I think what's going to be required is some new thinking in terms of what it is our visual system is trying to achieve. And I think what these experiments, so for example, what the size experiments are good for, is actually just challenging the traditional way of thinking about the visual system as a feed-forward hierarchical system, and remembering that feedback processes are going to have a large influence. But beyond that, we're going to have to be, I think, more creative in thinking about what it is these different visual areas are designed to do within the context of visual processing.
Thank you very much, Dr. Murray, for having time to speak with us today. We really appreciate it.
Oh, it was my pleasure.